Now, Carlos Heras and Daniel Reta is going to present us a new way of looking at reactivity, construction of a model from green fluorescent protein.
Okay, good afternoon. Thank you very much. And following the line of the two previous talks, we will be talking about, Carlos and I will be talking about green fluorescent protein. This protein is extracted from Aquaria Victoria, which is a jellyfish. And the importance of this protein is that it emits light. And the emission of the light, the responsible for the emission of the light is this little green molecule that is inside the protein. And as I said, is the responsible for the emission of the light. And based on this chromophore, we will propose a new way of looking at reactivity. First, some remarks on green fluorescent protein. The discovery and the development of this protein was awarded in 2008 with the Nobel Prize in Chemistry. And 6 years later, actually last year, 2014, the application of these previous discoveries to the field of bioimaging was awarded with the Nobel Prize for the development of super-resolved fluorescent microscopy. And basically, that's important because it's a matter of resolution. This new technique allows us to go further into details in bioimaging. And as a matter of fact, you can compare these two images, taken the uppermost image taken with conventional techniques, and the one below it with this new technique. And the improvement is obvious. Okay, but that's not really related to what we are going to talk about. And therefore, the question is, how is this related to cancer and malaria and reactivity? That is the topic of the talk. Well, in order to answer that, we need to explain first the proposed model that we are going to introduce here. But first, to do so, we need to explain this model. We have to first discuss the experimental evidence. And once we have done so, we will be in a position for asking ourselves again the question of how is this related to cancer and malaria, and also to answer, to have the tools to answer it.
So it allows us to explain how we met. Danny is a theoretical chemist who works in magnetic behavior of organic molecules, and I'm an experimental chemist who works at the same topic. But which is the starting point of our work? Well, at the beginning of my thesis, I was working about the study of nonlinear optical properties of a new kind of molecules based on two heterocycles joined by a nitrogen that shows fluorescence, curiously as green fluorescent protein. But during my PhD, we realized that it is not the only observed property. And this is what the talking is going to be about this new property. Well, but this starting point is too huge, too complicated. So we decided to simplify this system to a new, smaller molecule that shows the same scaffold and maintains the same property. So we got this one. So at this point, we have a new family of molecules, apparently neutral. But when we made the EPR spectra, we found out that they have this signal, and this signal has not supposed to be there, because this signal is not for neutral molecules. It is for free radicals. And we are not talking about free radicals, or maybe we are. And here comes the big question. Why is our molecule showing this kind of signal that is typical for a free radical? Well, radicals are very, very, very reactive species, and they are in degenerative processes such as aging. But our molecules, my molecules, have been stocked in my lab for months, for years, and they are still there. So in one hand, we have the radical properties, and in the other hand, we have very stable molecules. So at this point, is it that why we can, I call Danny, because we need a model to understand why this is happening.
Yeah, that's right. So basically, as Carlos just said, we got a signal that is not supposed to be there, so we needed a model to rationalize the experimental evidence. I'm going to be brief in this technical information, but I have to say it, so to explain the model. And the model that we propose is based on a conformational equilibrium between two species that have different multiplicity in its ground state. And the barrier to overcome, well, this barrier for this process can be overcome by thermal energy, and it involves basically a torsion of an angle. This might say nothing to you, but basically the message here is, and the message that you have to take into account is, keep in mind, is that through this torsion of an angle, it's accessible a species that shows a radical character and is the responsible for the EPR signal that we have measured experimentally. And so the extension of the model now it seems obvious, because since we have identified a structural pattern that is responsible for these characteristics, and this structural pattern is such a simple thing, we can ask ourselves, can we found this in other biomolecules? Is it a model that can be extrapolated to other kind of systems? And this is what we did. We started to look for them, and experimentally, if we find a structure that has this structural pattern, experimentally it should display the typical responses of a radical. So this is the point in which we link with the title of the talk, which is the green fluorescent protein, because as you can see by comparison, in this green molecule that I showed in the beginning, which is the responsible of the emission of light in the green fluorescent protein, it's very similar to the molecule that Carlos has been discussing, and it displays this structural pattern. So Carlos synthesized the molecule, we put it in a machine that measures the EPR signal, and we got this very nice signal. And this validated our model and promoted us to keep looking for other kind of systems showing the structural pattern. And in fact, we found some other molecules, and surprisingly, this molecule is dasatinib. Some of you might know it because it's the most sold anti-cancer drug in the market. And it displays here in red the very same structural pattern. So what we did was we bought the drug, and straight from the pot, we put it in the squid, which is the machine that measures this. And here you see this very characteristic signal of a radical. And not only this, we have also found in other families of molecules, such as a broad family for anti-malarial drugs, that this structural pattern is also found, and also in some neurokinase inhibitors, that are related with neurodegenerative processes.
Well, at this point, we have a lot of molecules with the same pattern that shows the same properties. And it gives us a new way of looking at reactivity that has been ignored until now, because they are more common than we know. And understanding these processes may help us in the drug design. So thank you, and
thank you.